was told I could talk. I was told I could talk about any topic that I teach on. So I chose plant reproductive systems because it's the area that it's closest to what I personally research. So for me, it's the biggest pleasure. So I hope you enjoy the topic as well. So in this lecture, I'll introduce a little bit the diversity of reproductive systems that you can find, particularly in plants, and they're amazing models for studying reproductive diversity. And then we will start to focus in on methods of outcrossing, since these are the most elaborate adaptations in terms of reproductive adaptations. And in particular, we look at one type of outcrossing mechanism known as self-incompatibility. And I'll take you through from the beginning, from the genetics of self-incompatibility through to some of my own studies in self-incompatibility. And then the later parts of the lecture, we look at how mating systems can evolve over time, and in particular, how they can change between selfing systems to outcrossing systems. So just a little bit of introduction about why outcrossing and mating systems are kind of advantageous in general. These ideas are date back really to the pretty early origins of modern biology and our beginning of our understanding of genetics. And so the great scientist Muller, as far back as 1932, posed this idea for why does sexual reproduction exist. And so he imagined a situation where you have populations that only self, that can't outcross, and they have different genotypes or genetic makeups. And if you have an allele that's a capital letter, it's an advantageous mutation. And so some of these lineages will outcompete the others and take over the space, but they can't gain any of the advantageous mutations that are in other lineages unless the mutation occurs within the lineage. And when that happens, that new lineage with extra advantageous mutations will spread and carry on taking over. If individuals could outcross and find other mates to cross with, if advantageous mutations that are in different lineages could come together a lot more quickly, and you would have recombination bringing them together, and the most fit offspring would be more rapidly produced. And so it speeds up evolution and acquisition of adaptive traits. There's also kind of general concepts how sexual reproduction promotes organismal complexity, and it allows more and more complex organisms to exist. So in terms of biology, what I mean by complex is that you may have more genes or larger genomes or extra genomes. And these extra genomes are usually associated with energy production, and energy production as a side effect can produce what are known as reactive oxygen species, which can be damaging chemicals inside the organism. But sexual reproduction can interact with these features of complexity and make them more tolerable. So sexual reproduction allows your genomes to recombine, we saw on the last slide. So you can combine favorable adaptations across different individuals. This recombination helps repair and purge deleterious mutations. It allows new adaptive combinations to come together, and so they can then spread in future, and all that new genetic variability. And so it becomes a virtuous circle that the more complex you become, the more advantageous it is to reproduce sexually, and that allows the accumulation of more complexity in turn. So in terms of plants, they have some particular challenges if they wish to engage in an outcrossing or sexual reproduction mode of lifestyle. Plants, of course, originally evolved in the sea, and in the ocean environment, there's a matrix to water to allow plants to disperse their gametes and their spores, and so they can spread and mix and reproduce in the water. It's a lot more difficult when you're on land. You're still sessile, so you can't move, but somehow those plants have to find another mate and exchange genetic material with that other mate. And so because of these challenges, plants have evolved a huge variety of mating systems to try and solve this problem. Yeah, so this mostly repeats just that. So plants can't move, so they still have the same problem as animals. They're trying to find another mate. And in terms of the diversity of mating systems that plants can show, they can either be inbreeding or selfing. I'll use both terms in this lecture, where the single individual is sufficient for reproduction, or they can show outcrossing mechanisms where you need a different individual, another compatible mate, in order to be able to reproduce. And plants are pretty good at both forms of reproduction, because the basic flower form is hermaphrodite. It's male and female. And so very often most plants have the option of inbreeding or selfing if they wish, because of this feature. So there are very many mechanisms to find that other mate if you are a sessile organism. And so the first option that you might have is to become an anemophilus, which means wind pollinated, and so to rely on the wind, on the air, to spread your gametes and spores. And so here's an example of a pine tree dispersing its pollen. And the wind pollination kind of syndrome, the characteristics that plants that are wind pollinated will tend to have are that their flowers will be, or reproductive structures will be very simple. They'll be exposed to the air, and they won't have petals or anything getting in the way. They'll produce very large quantities of pollen, and their female receptive organs, the stigmas, will be large and sticky as well. And so this is because, of course, you can't control where the wind blows, and so you need to produce a very, very large amount of pollen in the hope that some of it will land on the right species. Of course, we're very familiar with this other form of reproduction among plants, which is recruiting an animal vector to carry your pollen for you. And so we have very many entomophilous pollinated species, so pollinated by insects. And so plants have formed a lot of mutualistic relationships with many different groups of insects. And again, there's an entomophilous pollination syndrome that's typical of plants that might be pollinated by insects. So they need to attract insects, either with showy petals, various scents, nectar rewards. The pollen itself might be nutritious and might be the reward. And the floral organs themselves will tend to be very precisely arranged to promote cross-pollination. So we've got that whole group of plants. And further, there are more species that have become zoophilous. So they've developed mutualistic relationships with other organisms, such as birds or mammals. So here you see like a bat pollinating a night opening cactus here. We've got a bird pollinating this plant here, and they'll have their own specific adaptations depending on who's pollinating them. So if you're pollinated by something that's active at night, your flowers will probably be very pale or white. You're relying more on things like scent. If you're pollinated by birds, for example, birds see very well in the red color spectrum. So your flowers will probably be red or orange. Beaks, of course, lend themselves to being pollinated by tubular flowers. So the flowers will often form tubes as well. And this one is giving the plant kind of a landing platform as well. So we still have these, or typically have hermaphrodite flowers with male and female structures in the same organ. But if you're depending on outcrossing, you want to avoid selfing yourself too much wastage of your own pollen arriving on the female receptive organs. So there are various ways to promote outcrossing despite being an hermaphrodite flower. And so one of these is to show dichogamy, which is separation in time. So very typically, if you do have male and female organs in the same flower, they will mature at different times. And so you can have either protogyny, so that the female reproductive organs mature first, or protandry, where the male organs mature first. And we can see here in the image an example of aeonium with two flowers of different ages. And the younger flower has showing protandry, so the stamens here are receptive, they're releasing their pollen, they're pointing forwards. The female reproductive is still immature and still all closed up, so it's resisting being pollinated here. And then in this older one, the flowers carried on developing, it's pulled back to stamens that are now expired, and the female stigma has carried on developing and has opened up and will receive pollen from other flowers. In addition to separation in time, we can have separation in space, which is called hercogamy. And so again, this is about separating the male and female organs within the flower in such a way that it discourages self-pollination but encourages cross-pollination. And so here's one typical example. The lengths of the floral organs might differ. And so if you show approach hercogamy, you might have the female organs are longest. And so if insects are arriving from the front of the flower, they'll come in contact with the female organs first. If they have to be carrying pollen from other flowers, that will hopefully transfer at that point. And then they'll come in here for the rewards and they'll pick up the pollen. And you can have the reverse situation as well, reverse hercogamy, where it's the male organs that the insect comes in contact with first, and later it'll come in contact with the female organs. And here you can see another typical example, again, like we saw in the previous image, that as flowers carry on developing, the floral organs will actually move around inside the flower and move in and out of the way. So as this flower is developing, the expired anthers are pulling back, and, so that they avoid pollinating their female stigma. An interesting form of hercogamy is known as heterostyly. And so this is where you have kind of approach hercogamy and reverse hercogamy in the same species, in the same population. And so we have some individuals in the population will have long female organs here in the green and short male organs. And other individuals will be the opposite arrangement. They'll have short female organs and long male organs. And so this makes cross-pollination more efficient between the two floral types, because an insect entering this floral tube will tend to pick up pollen towards its head in the short male form and towards its tail in the long male form. And that pollen is at the right height to cross-pollinate the opposite female form. So there's typically two flower forms in heterostylous systems. And here you can see that as a cartoon again. So an insect visiting these flowers will be picking up pollen in one part of the body in one flower form, and that part of the body is ideally suited to the opposite flower form, where it'll cross-pollinate, and vice versa. Another way to ensure outcrossing is to become either more completely male or female. So either the flowers become completely male or female, and they can still be on the same individual, but they're separated in separate flowers now, or the whole plant can become male or female. And there are plenty of examples of both of those kinds. If the whole plant is male or female, that's the most extreme form, because of course you're an obligate outcrosser at that point. You have to find another individual in order to reproduce. And the males, I guess, are offsetting the cost of not being able to produce seed by being able to pollinate many more females in that population. And at a genetics level, Daiichi at its most extreme will have entire chromosomes that are devoted to determining whether those individuals are male or female. So they'll have evolved a whole sex chromosome. And plants are really useful and really interesting for studying sex determination, because it's a trait that's evolved multiple times among plants. And we have a kind of all different stages in the evolution of sex chromosomes, all the way from hermaphrodites through to the most extreme of the male and female plants with sex chromosomes. So we can kind of capture evolution in action. So here's an example of that. The genus Silene, the campions, has been extensively studied for sex chromosome evolution. And that's because it's evolved recently within this genus in the last few million years. And also, when you build evolutionary trees for this genus and you map mating systems onto it, you can see the outgroups here. The ancestors were hermaphrodite. And then within the genus, we've got a mix of purple groups here, gynodioecious. This is a mating system where you have females and hermaphrodites in the population. And then you have fully dioecious species. So these have males and females. So you can see how males and females tend to evolve from systems of females and hermaphrodites, which in turn have evolved from the hermaphrodite systems. And this tree also shows you that there are two origins of dioecy, of sex chromosomes, just within the single genus. And they were independent evolutions and involved different chromosomes. So we can learn a lot about what's a unique event, what only happened once, and what's shared across systems that evolve sex chromosomes. Okay, so I'm going to develop one type of mating system in a little bit more detail, because I have more personal research experience of it. And so this mating system is called self-incompatibility. So what self-incompatibility is? Well, for a start, it's a very common outcrossing mechanism. So around 39% of all flowering plants show self-incompatibility. And it's the ability to recognize and reject self. So the female reproductive tissues are able to recognize when their own pollen and reject their own pollen, but accept pollen from another individual of the same species. And genetically, it's very interesting, because it tends to be controlled by what's known as a supergene. And so that's a genetic locus that has at least two genes, one that's expressed in the pollen, and the other one that's expressed in the female reproductive tissues. And these are interacting specifically for the self-incompatibility interaction. And that self-incompatibility interaction, although it's evolved independently multiple times, it always shares these features in common. And so one of them is this lock and key mechanism. So those male and female gene products are specifically interacting with each other. And when the lock fits, it activates self-incompatibility and pollen rejection. So the males are basically signaling, and the female receptors are looking for the specific signal of self, and that will cause the pollen rejection pathway. And if there's no recognition, that pollen isn't recognized as self, and pollination can proceed as usual. And so this explains why it needs a special supergene structure of two different genes that are linked in the genome, because that interaction needs to be specific. So you can see it here shown as a cartoon. So here are the male and female expressed genes. And the alleles of these genes interact specifically with each other. So the two genes that are found on this haplotype will only recognize each other. So you can see by the shapes here. And a different allele, a different S allele, will have different specificity that only these two that are on the same haplotype will recognize. So again, shown here by the triangles. And so the need to keep the matching pairs of male and female alleles together is usually solved by placing them next to each other on the genome so they're not broken up by recombination. And here you can see again another cartoon kind of showing self-compatibility in action. The pollen grain is expressing those male genes, circles and triangles. The female stigma surface is expressing these the female S genes, the receptors. And because in this example it's a self-pollination, these are triggering recognition by the female receptors and activating a pollen rejection pathway. So it's very important for the function of self-compatibility that those male and female alleles stay together on the same haplotype. If they separate, you won't be able to have self-recognition anymore, and self-compatibility will break down. So beyond being physically close to each other, there's still a small probability that recombination can take place there. But to reduce that probability further, we find that the S locus tends to be very structurally variable. And so here you can see examples, cartoon examples of different S alleles that have been sequenced in this species, Arabidopsis lyrata, and showing the position of the respective. This is the female genes, SRK, these are the male genes, SCR, and the numbers are referring to different alleles that have been identified. And you can see that within this genome region, the location of the genes varies, and also sometimes the direction of translation varies. And so there's a lot of structural variation here, and that's inhibiting these different S alleles lining up very well during cell division and during meiosis. And if they're not lining up, they can't recombine. So it's protecting this genomic region from recombination and making sure that the male and female genes tend to stay together and are inherited as that single unit, that single supergene. In terms of kind of controlling selfings, there are also different types of self-compatibility that offer greater or lesser degrees of control of mating with relatives. So self-compatibility systems, because they're based on recognition of shared S alleles, your relatives are more likely to share an S allele with you than an unrelated member of the population. So it also serves to indirectly reduce inbreeding by mating with your relatives. And so if you express something known as gametophytic self-compatibility, that means that the male gametophytes are expressing their own male S allele. And so because the male gametophytes, the pollen, are haploid, they only have one allele that they can express. So each individual pollen will express a single S allele that was present in the father. The female tissue is sporophytic, it's diploid, and so it's capable of expressing the two diploid alleles that are present in the mother. So in this example, the individual is expressing alleles 1 and 2. So this tissue is expressing recognition factors for 1 and 2. But the individual pollen will either be 1 or 2. And so this is an example of a self-cross where the individual was originally S1, S2. And so all pollen will be either S1 or S2 and will all be recognized by the S1, S2 female tissue. If it's an unrelated individual, say the father was S3, S4, all pollen will express either S3 or S4, neither of which is found in the female tissue, and so it won't be rejected and it will fertilize. But if you are related, for example, you're a half-sib, you might share a single S allele in common. So in this example, you might share S1 in common. And so only pollen that expresses the S1 allele will be rejected. The other one, S3, that isn't found in the female tissue, will still germinate and grow. So you get half compatibility there. So you get partial protection against relatives under a gametophytic system. If the male S alleles are expressed earlier during development, so in the stamen and by paternal tissue, this paternal sporophyte tissue that's diploid, that means that the individual pollen grains will be coated with both the male S alleles of the sporophyte father. So each individual pollen grain is showing S alleles for the two S alleles of the paternal parent. And here in the case of crosses between relatives that might share a single S allele, because every individual pollen grain is now expressing both the S alleles of the father, they're being recognized by the S1, S2 tissue, and there's full incompatibility here. So there's more effective protection against crossing with relatives under a sporophytic system. So self-compatibility has evolved independently multiple times, and as we've just been describing, the things that are shared is that they all have this supergene system with male and female expressed genes, and they can either be expressed, the male tissues, the male S alleles can either be expressed at the sporophytic or at the gametophytic stage. Gametophytic systems are fairly common, and there are multiple different kinds. So here are two of the more common kinds. In this system, you have the male gene happens to be the receptor, and the female one is the signaling molecule. And so the receptors are on the surface of the growing pollen tube, and if they interact with the female signaling molecules, it will trigger a calcium signaling pathway that will lead to the destruction of that pollen tube. In this other common sRNA system, again, the pollen factor is an inactive sRNase, and there's a signaling female molecule that enters the growing pollen tube freely, and if it interacts in a specific way with the sRNase, it will activate it, and the pollen will destroy its own RNA, and that will kill it. That's the incompatibility action there. In the only sporophytic system that's known at a molecular level is the Brassicaceae system that we've seen examples of already. It's the female gene is the receptor, the male one is the signaling molecule, and so these receptors are on the surface of the stigma, and if the pollen signaling molecules specifically activate the receptors, that triggers phosphorylation and a rejection pathway. In terms of the evolution of self-compatibility systems, they evolve in fairly unique ways, very interesting. And so one feature is that it's very difficult to evolve a new specific S allele specificity, because typically evolution takes place one mutation at a time. And so if you have a mutation that changes the specificity of either the male or the female partner, it will no longer be recognized by the other partner that hasn't changed yet. And so this allele will actually be inactive. It won't recognize self anymore. And so the self-incompatibility system will be dysfunctional. And it has to wait until there's another mutation that restores a new unique specificity before this becomes a new functional S allele. So this is a very rare event, and so new S alleles evolve only very slowly. However, it's balanced against the fact that new S alleles are very selectively favored. They're very advantageous, and that's because you've got something called negative frequency dependent selection acting at the S locus. And what that means is the rarer you are, the more fit you are. And you can see here in the cartoon how that works. If you have, so the different colors represent individuals expressing different S alleles. And so if you share a color, you share the same S allele. And so in a population with few S alleles, you won't be able to mate with yourself, but you also won't be able to mate with anyone else in that population with whom you share the same S alleles. These could be your relatives, or they just represent not very much S allele diversity in that population. And so the proportion of individuals that you can mate with, that's your mate availability in that population. But if you're a rarer S allele, so in the example here on the right, you can actually mate with a much bigger proportion of the population. Your mate availability has gone up, and so chances are you'll be more fit on average, and so the frequency of your alleles will increase in future. And the equilibrium situation under negative frequency dependent selection is that however many S alleles there are in the population, they'll all be at more or less equal frequency. But you can also see why this is an important driver of new S allele diversity. So if you do manage to evolve a new S specificity, a new S allele, you'll be strongly favored when you're rare, and you'll spread in that population, and you'll also be favored to spread between populations by migration, because if you're a new S allele arriving in another population, again, selection will favor your establishment and spread in that population. And so the net balance between these two forces, on the one side, that it's very difficult to evolve a new S allele specificity, and on the other side, when those new S specificities do evolve, they're highly fit and they tend to be maintained, is that over time, S allele diversity, over evolutionary time, S allele diversity will gradually increase and become very, 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 very polymorphic. And so the S locus is the most polymorphic S locus, polymorphic genetic region known in plants, and similar to the diversity that you find at the immunity genes in mammals, for example, the MHC genes. And so how that might look in terms of the evidence is if you build evolutionary trees of S genes, they'll have these characteristics. They'll have very long branches leading to the S genes, reflecting their great age, that they've been maintained over long evolutionary periods. And the colors in this particular tree refer to the species that the S alleles were sequenced in. And the red and blue, so this lower part of the tree, came from the same genus. And you can see there's extensive S allele sharing between these two different species. And this, coupled with the long branches, says that these genes are older than these species. They've been maintained over very long evolutionary periods. And you can also see that in terms of some of the allele pairs between species are closely related to each other. So they're basically equivalent S alleles that have been found in each of those species. Up here, we've got a species in a different genus, Brassica versus Arabidopsis. And even here, there's some S allele sharing, showing that some S alleles are even older than the genus. So very, very ancient S alleles being maintained over evolutionary time here. Okay, so I'm going to talk a little bit about my own research in self-incompatibility and how it evolves. So the system that I worked with for my PhD are the ragworts, the senecio, and the particular species that I worked with where there's an endemic species called Etnensis that grows at the top of Mount Etna in Sicily. And it comes into contact with a closely related species, Chrysanthemum folius, that exists only at lower latitudes, and both species are self-incompatible. Where the two species come into contact at middle latitudes on the mountain, they form a natural hybrid zone. And this species is of interest to UK researchers because these hybrids were introduced to the UK about 100 years ago to Oxford Botanic Garden. And the hybrids became a successful invasive species over the next 300 years. And so it's spread throughout the UK. And it's an urban weed. It spread initially via railways, and it's now found in most major cities in the UK. So it's of interest in that sense. In terms of self-incompatibility, all of these species are self-incompatible. So we can ask questions: how does self-incompatibility interact with hybridization between closely related species, and how does it interact with invasive evolutions, becoming a successful invader? All these species belong to Asteraceae, and it's a sporophytic system. And so we don't actually have the molecular details of this system yet. But you can still study S alleles by their function. So you saw here how, in previous examples, how if you have one individual and you perform controlled crosses with other individuals in a sample, if they're compatible, then you know that the S alleles they express were different. And if they're incompatible, you know that they share the same S allele. So you can perform what are called diallel crosses. You perform every paired combination of crosses in a sample and figure out which were the compatible interactions, which were the incompatible interactions, which were the incompatible interactions. And from that, you can figure out what S alleles were present in that population. So here's what that might look like. Each square in this table represents the outcome of a paired cross between the individuals listed along the top and along the side. And so the negative was an incompatible reaction, the positive was a compatible. The greens here were an undecided interaction. And so the individuals have been ordered to group incompatible individuals that are all interacting with incompatibility together into an incompatibility block. And so all these individuals here share the same expressed S allele. And so we can start to learn about the S allele diversity of populations from these results. So you can count the number of incompatibility blocks to find out how many S alleles there are in your population. You may learn about dominance interactions between S alleles. So this individual, for example, expresses 2 S alleles as one sex, and only one S allele is the other. And so you can do that then for well, in my case, for different species and for different populations to understand about their S allele diversity. So what you just saw was a result for the UK invasive species. On this page, you can see results for populations from the Sicilian species. I don't know if you can see it already, but the incompatibility blocks are smaller and there are more often on Sicily. So we have greater S allele diversity in the native range than in the invasive range. And also we can see that the hybrids show high S allele diversity similar to the 2 parents. So there's lots of S alleles entering this hybrid zone and mixing between the 2 species. So here you can see a summary result of that. So on Sicily, how many S alleles did have, what was the frequency of different S alleles across populations that I found, and many of them I only found once in a single sample population. And when you compare the different species and the hybrids from Sicily with the UK populations, you can see that there's much less S allele diversity in the UK populations. Here it is as a figure, reflecting a population of bottleneck associated with invasion. However, when you convert that to mate availability measures, so the average proportion of individuals that any one individual can mate with, you can see that although there was a large reduction in S allele diversity, there's a less extreme loss of mate availability. So on average, a UK individual can mate with 80% of the other individuals in a population, compared to more than 90% in Sicily. So there's a slight fitness cost, but it's not huge. And so probably it's this that has allowed this invasive species to maintain its outcrossing system, even though it is an invader. And here's another impact as well. We saw in the previous graph, whereas alleles in Sicily are rare, and I tended to only observe them in single populations. The relatively few S alleles that we saw in the UK ones are present in nearly all of the populations. There are few S alleles that they're under strong selection to be maintained across all of those populations. Okay, so that leads me on to the last part of the lecture, which is mating system evolution. When do mating systems evolve? So you heard me talking before how we expected that there was pressure for the mating system of the UK invader to have changed as part of becoming an invasive species. And although there were some changes, they hadn't changed as dramatically as we expected. And the type of changes that we often see when mating systems change is a switch from outcrossing to selfing. That's a really typical one. And so you can see here in cartoon form, well, it's an evolutionary tree of a genus, Collinsia, that shows a mix of outcrossing and inbreeding mating systems. So where you see a large flower, it's an outcrossing species. Where you see small flower, it's selfing. And you can see a lot of closely related outcrossing selfing pairs here, but the relatives tend to be outcrossers. And so that's reflecting the fact that there's a frequent switches from outcrossing to selfing over evolutionary time, but also that they don't appear to last very long. They tend to be quite young changes. It's not the only switch that can happen. Rarely you can have switches from selfing to outcrossing. So here's a very striking example in Juncus, a sedge. They're typically wind pollinated, and rarely you find species that have developed insect mutualisms. And so they're beginning to evolve all the characteristics of insect pollinated flowers again, so showy flowers again. So at the beginning of the lecture, we discussed why some reasons why outcrossing is advantageous. It generates new genetic combinations, and it allows for recombination to share beneficial mutations and to purge deleterious mutations. So these are great evolutionary advantages. But of course, selection works on the here and now. Are you fit in this moment? And so the short-term advantages of outcrossing are probably that within individuals, you're more variable. So you show more heterozygosity. And those heterozygotes are helping mask any genetic load that you might have. Any deleterious mutations are masked with a fully functional heterozygote allele. And so that helps avoid inbreeding depression. And empirically, we can see these results. So this is a measure of genetic diversity, and is comparing these two closely related species, an outcrosser and a selfer. And you can see the outcrosser shows greater genetic diversity compared to the inbreeder across these two different measures of genetic diversity. And that's because selfing causes you to lose heterozygosity very quickly. So even if you start heterozygous, as you can see in this Punnett square, and your gametes can either be A and B, if you're selfing and these gametes are reuniting at random, half of your offspring will be homozygous. And that happens at a single locus, or when that's happening across your whole genome, you're doubling your homozygosity every generation of selfing. So you rapidly become very highly homozygous. And so when you're homozygous, you are not hiding your genetic load anymore. And so all these mildly deleterious alleles that might be hidden in heterozygous state are now expressed. And so that they'll manifest as what's known as inbreeding depression, lower fitness of self to individuals. Selection can act against this, and so you can gradually remove these deleterious alleles. That tends to happen in some selfing species, that they have purged their genetic load. But there's a race here, and you survive the lower fitness until your populations have purged all the genetic load. But the reason why their evolution seems to favor the evolution of selfing so easily is these two kind of linked concepts: mate availability and reproductive assurance. And so here you can see this example of, say, a self-incompatible species becoming self-compatible. So before, you see that you can only mate with individuals that you're compatible with, whereas if you're self-compatible, you can actually mate with everyone in the population, plus you can mate with yourself. And reproductive assurance, imagine you're founding a new population, or your population is small or low density. You might not find any mates, but if you're self-compatible, you can always have the option of mating with yourself. So you've got that reassurance. And so anything that threatens either mate availability or reproductive assurance can trigger, it can be a selective force towards selfing. And so you've got lots of ecological factors, reductions in population size or density. You occur at the range margin, you're a colonizer, environmental stresses, fragmentation, and so on. All of these can favor a switch to selfing. And so the grayed out ones here show rare reproductive shifts. So it's rare for selfing to shift to outcrossing, where it's common for outcrossing to shift to selfing. And it's common for new selfing species to bud off from outcrossing species, for example. And in terms of diversification, new species of outcrossers will tend to be sexual conflict that's preventing hybridization, whereas for selfers, it's simply isolation because they're not as well connected. They'll tend to evolve neutrally and take unique evolutionary trajectories. And becoming extinct is you lose ability to outcross in outcrossers, whereas in selfers, it's the accumulation of inbreeding depression and deleterious mutations. And this is the last slide. So you can model all those processes to get a feel for why do modern plants show a mix of outcrossing and selfing. And so the data that you put in is the lengths of branches on trees and whether they are selfing or outcrossing that particular species. And so then you can model here self-incompatible to self-compatible, the rates of mating system shifts, and the rates of speciation and the rates of extinction. And you can run models with different parameters and ask, do they produce a tree like the one that we actually observe? And on balance, what you find are these rates that selfers might evolve more quickly, but they also go extinct more quickly. whereas outcrossers might evolve more slowly, but they tend to persist for longer. And it's this equilibrium that leads to such mating system diversity over evolutionary time. Okay, so these are just the summaries that, of what I said, how sex goes along with complexity. Plants show great diversity of reproductive structures, and then we listed various kinds of outcrossing mechanisms, such as dichogamy, hercogamy, and so on. We went into more detail about self-incompatibility, the various different forms that have evolved, the type of evolution that happens at the S locus with some examples. And then we looked at typical patterns of mating system shifts and the various evolutionary drivers that tend to cause them. Okay, thank you very much. And I'm happy to take any questions.
Thank you very much, Adrian, for your very interesting lecture. And, шановні учасники, можливо, в когось є яке запитання. Якщо є запитання, можете вмикнути мікрофон та задати. You know, Adrian, it was very exciting to see then in hybrids the as alleles diversity is more than in initial species. And how do you think, is it for all species are common, or maybe it's just some particular case?
Yeah, I suspect there's an equilibrium, because self-incompatible species are not only are they better at recognizing self, but they also seem to be better at recognizing foreign pollen. So they are good at limiting hybridization, but you also have it being balanced by the fact that new and rare alleles spread between populations more easily. So I think it all depends how closely related the species are. If they're very closely related, it will promote more hybridization and the species kind of mixing together more. And if they're a little bit more distant, it becomes a very effective barrier to hybridization. So I think it can work both ways.
Okay, because you know, it's a very interesting question, especially in natural conditions. Now we have the globalization of the even people. It's rise to globalization of the maybe some migrations of the pollen, of the seeds, of the spores. So now we can detect more and more globalization of species, and then they can make some hybrids in new regions, in new areas, and it is very interesting how it will be on the level of the genetics of these alleles, and maybe some new alleles even can from in other conditions, it's like mutation factors for the species, and they can produce new alleles, new variants of alleles, and maybe it sometimes can be helpful, and maybe sometimes I think it maybe even will hardness for the species, because I think it's a very real situation when the species is in new conditions, and if they was not do not have before a self-incompetence in these regions, it can be shared. And Adrian, we have some question in the chat. Self-incompatibility does not affect the distribution of plants. If plants cannot self-pollinate it or use related pollen, how do they reproduce in nature in sufficient quantities? No human intervention.
So is that question specific to self-incompatible species? Do I understand rightly?
Yes, I think yes. Dasha, it's for self-incompatibility.
Okay. So I think if I understand the question properly, you're referring to there are situations when a self-incompatible species will have problems reproducing if it's in a new area. Is that right?
I think it's how they can maybe cope with the problem when they cannot self-pollinate it, or if they in the regions where they not have a related pollen species. So,
okay, yeah.
mhm.
So definitely that's one of the pressures to evolve towards self-compatibility. So to lose your self-incompatibility. Well, what can happen rather than completely losing your self-incompatibility is that many species show what's called mixed mating. And so although there's a preference for outcrossing when there are pollinators and other mates available, if there's no option, they will then be able to self and produce some self seeds. And so it may be that could be a solution as well, that plants will temporarily self when they have to, but if, as soon as the population recovers and they have pollinators, the self-incompatibility will return.
Okay, thank you. And other questions do you have in the audience? Ще є запитання в аудиторії присутніх? Ну, якщо запитань нема, тоді, мабуть, ми будемо дякувати пану Адріану і відпускати його. So I think maybe the questions is finished. So